Hello, everyone, and welcome to today's EBD Group Bioeconomy webcast titled NAMs Empower Translational Research, sponsored by Axcelead Drug Discovery Partners. This is part 2 of our 3-part series titled Transform Your Drug Discovery, Faster Paths to Clinical Success. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, you can submit your questions into the Q&A window on the left-hand side of your screen. And in 24 hours, you'll receive a link to watch the recording of this session. Let's now begin by introducing our first speaker today for this session. We have Masayuki Goto. He's the head of the Pharmacology Business Unit for Axcelead Drug Discovery Partners Incorporated. Welcome, Masayuki. Please begin.
Okay, good morning, good afternoon, and good evening, where you are joining from. Thank you for the introduction. I'm Masayuki Goto from Axcelead Drug Discovery Partners. It's a pleasure to be here today. In this webinar, NAMs Empower Translational Research, I will share how we are applying new approach methodologies to improve translational predictability in drug discovery. Before we begin, let me briefly introduce our company. Axcelead is a Japanese integrated drug discovery solution provider, founded as a spin-off from Takeda and Teijin. We operated 2 sites, Shonan and Hino, with over 300 scientists, many from the pharmaceutical industry. Our compound library exceeds 1.2 million molecules, and we've accumulated data from more than 1,000 drug discovery projects. With over 290 clients and 90% repeat rate, our track record reflects strong trust and consistent quality. We also collaborate with global pharma companies like Lilly and Acadia and have achieved an 84% success rate in lead generation and optimization. Axcelead contributes to the client drug discovery challenges through a pharma-grade platform. We provide a comprehensive integrated drug discovery service from target identification and validation to IND enabling studies. Our scientists bring deep pharmaceutical expertise, and we leverage legacy data combined with proprietary AI to boost innovation and a success rate. Our platform supports a wide range of the modality, including next-generation small molecule, peptide, antibody, and oligonucleotide, enabling seamless collaboration and continuous improvement. By integrating people, technology, and data, we accelerate drug discovery and delivery highlight value to our client. Okay, let's move on to the core of today's presentation. I will introduce the key platform that forms the foundation of our NAMs-based approach. NAMs are innovative technologies aimed at improving the relevance and predictive power of preclinical research while reducing reliance on animal models. To support this, FDA published a roadmap out-license technologies expect to shape future preclinical testing. This innovation landscape, including some technologies shown here, all designed to enhances translational predictability. At Axcelead, we are contributing to this global movement by developing platforms aligned with NAMs principles. In today's webinar, we will highlight 3 major platforms. I will present our human tissue-based systems, and Dr. Satomi will discuss multi-omics, and Dr. Jason will introduce immune-humanized and MPS. Together, this talk will show how we are responding to the NAMS roadmap and advancing translational research. Now that we introduced the overall structure of today's webinar, I'd like to move on to my session. My presentation is titled Human Tissue-Based Evaluation Systems, and I'll be covering three main aspects of this platform: human tissue-based system, organoid platform, immune-humanized mouse model. These platforms serve as bridge between preclinical research and clinical success, enhancing translational relevance. Now let's dive into the first topic, human tissue-based evaluation system. Fresh human tissue provide a valuable opportunity for translational research. They allow us to validate drug efficacy beyond cell line and animal models, detect human-specific effect, discover novel biomarkers and therapeutic targets, and reduce reliance on animal testing. However, access to fresh human tissues suitable for functional analysis is limited, often due to lack of the clinical context and standardization. To address this, we collaborate with HAB. These partnerships enable timely translatable acquisition of fresh human tissues from local hospitals, supporting physiologically relevant functional assay. Our platform enables rapid processing of the fresh clinical samples, typically within 2 to 6 hours after surgical resection. This ensures minimal degradation and maximal biological relevance, which is critical for accurate functional analysis. We collect detailed patient information, including age, gender, drug history, and genetic mutation. We collaborate with multiple partner hospitals that provide a wide range of high quality traceable human samples, including tumor and normal tissues and biofluid. These include fresh isolated samples such as bone marrow, liver, adipose tissue, skin, and small intestine, enabling fast and reliable data generation for translational research. Fresh human tissue provide a physiologically relevant platform for evaluating drug responses, preserving cellular diversity and immune context. We process tumor and normal tissues using enzymatic dissociation method. In particular, our 3D culture called tumoroids contain cancer cell, cancer-associated fibroblasts, immune cells, epithelial cells. We also apply a similar approach to normal tissue to study rare immune cells population and specific resident cells. In the next slide, I will show a case study using colorectal cancer derived tumoroids. As introduced earlier, our 3D tumoroid culture preserves the cellular complexity of human tumors. In this case, we use fresh colorectal cancer samples to generate tumoroids containing cancer cell, immune cell, and CAFs. Importantly, these tumoroids also retain the tumor-infiltrating lymphocyte TILs, allows us to evaluate drug responses in a physiologically relevant microenvironment. We performed a viability assay to assess therapeutic efficacy, demonstrating how this platform supported human relevant oncology research. In this case, we focus on the normal colon tissue to isolate it and analyze rare immune cell subtypes. Using flow cytometry and cell sorting, we achieve high quality, high purity over 90% is isolating target immune cells. We then perform functional assay to evaluate proliferation and cytokine production. Fresh tissue is essential for maintaining the physiological environment of immune cells, which is critical for accurate analysis. Together with the tumor case, this example highlights the versatility of our human tissue-based systems. We've just explored how fresh human tissue can be used to build physiological relevant platform. Next, I will introduce our organoid platform. Our brain organoid platform provides physiologically relevant models for studying human brain tissues. These organoids are reproducibly generated with a layer structure. You can visually observe this layered organization through immune staining. We perform phenotypic assay to evaluate protein aggregation, which is essential for modeling neurodegenerative disorder. In addition, we assess neural activity and disease-related phenotypes, including MEA recording. Further detail about MEA will be presented by Dr. Jason later in this session. These models offer a robust platform for CNS disease research. Using our brain organoid system, we've developed a model for Alzheimer's disease. By treating organoids with A beta oligomer, we observed clear accumulation of amyloid beta, one of the key pathological features of the disease. These results demonstrate that our organoid model reliability replicates neurodegenerative phenotype. This approach enables both drug screening and mechanistic studies in a human-related relevant context. That concludes our overview of our human tissue-based platform, including fresh clinical sample and brain organoids. Next, let's move on to our immune-humanized model, humanized mouse model for in vivo assessment of the immunotherapies. Our immune-humanized mouse models are designed to evaluate immune-targeted therapies in vivo. We use both PBMC-based and HSC-based approaches and apply hydrodynamic gene delivery, HDD, to express human cytokines like IL-15. Hydrodynamic gene delivery enables rapid transient gene expression by injected plasmid DNA into the bloodstream under high pressure. This supports engraftment of immune cells, including T cell, B cell, NK cells, macrophage, and plasmacytoid dendritic cells. Depending on the model, we can achieve short or long-term immune cell reconstitution tailored to specific therapeutic needs. We use HSC-based HuNOG models with hydrodynamic gene delivery to transiently express human IL-15, which is essential for NK cells. In standard HuNOG models, HSC transfer typically result in the engraftment of T cell and B cell only. However, by introducing IL-15 by HDD, we successfully achieved robust NK cells engraftment. This model is ideal for studying chronic immune responses and assessing the efficacy and safety of immune therapies. Okay. This is final slide. In conclusion, our integrated platform, fresh human tissues, organoids, assays, and immune-humanized mouse model, a powerful tool for translational research. They align with NAM's principles and enhance the predictive power of preclinical studies. By bridging the gap between bench and bedside, we aim to accelerate development of effective and safe therapies. Thank you for your attention.
All right, Masayuki, here's our first question for your section.
Okay.
How do you ensure the quality and consistency of fresh human tissue samples across multiple hospitals, especially when expanding your network?
Okay. Thank you. That's an important point. As we expanding our hospital network, we place a strong emphasis on maintaining sample quality. We work closely with pathology teams at each hospital who visually inspect and select high-quality tissue sections. This collaboration allows us to receive the functionally variable samples with minimum delay, typically within 2 to 6 hours post-surgery. Their expertise ensures that the samples are suitable for downstream assay, and we continue to refine our protocols to maintain consistency assay across site. Thank you.
Many thanks, sir. Next question here. How do you handle variability in human tissue samples when conducting functional assays?
Thank you. That's a very relevant question. While variability is inherent in human derived samples, we've found that drug responsiveness can be consistently captured across different sample lots. This is largely thanks to our close collaboration with hospital pathology team, who visually inspected and select highly quality tissue section. Combined with rapid processing and standardized assay protocols, this approach allows us to maintain functional viability and reproducibility. So even with biological diversity, our platforms reliably detect pharmacological effect in a translational, meaningful way. Thank you.
Thank you, Masayuki, for an excellent segment. Let's now introduce our second speaker. We have Yoshinori Satomi, PhD. He's the director for Omics and Bioinformatics Group at DMPK Business Unit for Axcelead Drug Discovery Partners Incorporated. Welcome, Yoshinori. Please begin.
Okay, so thank you so much for the opportunity to introduce our Omics technologies today. My name is Yoshinori Satomi from the Omics and the Bioinformatics Group at Axcelead. Today, I'd like to share with you our approach of multilayer omics technology and how these technologies can significantly contribute to translational research. Today, I will cover 4 parts. First, why omics is important for translational research for new approach methodologies as well. Second is overview of our multilayer omics technologies. And then I'll cover service detail and case studies. Finally, I'll summarize the key message of this presentation. So here, I'd like to highlight the importance of omics in the context of NAMS. As you know, NAMS aims to replace animal testing with more human-relevant models. However, there are still challenges that are predicting human drug responses, and the mechanism remains difficult with these models alone. They provide variable phenotypic information, but not always the molecular details we need. Applying omics data drug evaluation process, we can capture molecular level insight into efficacy and toxicity, adding mechanistic depth and human relevance. And most importantly, it facilitates translational research, bridging the gap between the preclinical model and the clinical application. So let me introduce our multilayer omics technologies here. At Axcelead, we provide an all-in-one platform that enables comprehensive acquisition of omics data across multiple omics layers, metabolomics and lipidomics, proteomics, and transcriptomics and genomics, as well as some applied solution taking advantage of our capabilities. This integrated approach supports a wide range of applications in drug discovery, such as biomarker discovery, target identification, and drug repositioning as well. So in the next section, I'd like to show the example of omics analysis by dividing into three parts. The first topic is omics-guided mechanism-based efficacy. So as you know, efficacy observed in clinical models often does not fully translate to human patient. One way to bridge this gap is understand the molecular mechanism underlying compound action. Omics analysis provide a comprehensive system-level view of how compound perturb the biological pathway and the networks. By linking the mechanism of action directly to observed efficacy, we can strengthen the rationale for drug development decisions. This approach not only guides rational compound selection, but also identified candidate biomarker that can predict efficacy and help stratify patient population and improve the translational value for clinical findings. This slide, I'd like to introduce our evaluation process for nucleic acid medicine using transcriptome and genome-based analysis. We first conduct an in silico analysis to predict off-target genes by comprehensive sequence similarity against human genome using our optimized sequence search algorithm. Then obtain in vitro transcriptome data following the treatment in relevant cells. These data then combine to narrow down off-target genes candidate and assess the risk based on the officially provided gene, risk gene data and the phenotype gene relation databases. So this slide shows example of transcriptome data for 4 antisense oligonucleotides. 3 of them are already approved drugs, and one was halted at phase 1A. The green dot indicate the target genes whose expression levels are changed as expected, which means confirming the target engagement. These red dots represent genes that are significantly altered aside from the target. For the approved ASOs, the number of these differentially expressed genes is very limited, suggesting the minimal off-target effect by this drug treatment. So by simply comparing gene signatures, we can extract a great deal of information beyond the target engagement. Moreover, by integrating the in silico prediction data, we can more accurately identify the potential off-target genes. This combined approach enables us to perform a simultaneous assessment of efficacy and toxicity. So this is a case study of evaluation of VEGF-A targeting ASOs. So transcriptome profiling showed all that candidates successfully knocked down VEGF-A, but each displayed distinct off-target signatures. So differential gene expression signature highlighted VEGF-A number 1 as achieving the best balance between on-target potential and the minimal collateral pathway effect. And principal component analysis of these data further separated their trend. this kind of analysis allowing us to explore molecular mechanism of action in terms of both on-target and off-target effect. So this type of evaluation approach can be applied to the other modalities by using proteomics and metabolomics as well, where molecular signature provide more biologically relevant insight. So next topic is the discovery of disease mechanism by omics analysis. So non-target metabolomics is a powerful approach to discover novel disease mechanism by capturing unexpected changes. Our platform integrates LCMS and unique search algorithm and large annotated molecular libraries. The key strength is the extensive molecular database we have built by systematically acquiring the past research findings. So this database includes the virtual structure library predicting the diverse lipid structures across more than 500 lipid species. This capability allows us to reveal disease mechanism that conventional approaches cannot identify, providing a strong rationale for translational research and drug discovery. This is an example of non-target metabolomics for a renal carcinoma case. In this study, we identified unique lipid signatures, such as very long chain polyunsaturated cholesterol esters, and also ACL-linked glycerols found only in malignant lesions. These specific fingerprints provide a new insight into renal carcinoma biology and may inform therapeutic strategies and patient stratification ideas. And our next platform is proteogenomics. This approach creates a disease-specific protein database, sequence database, informed by disease RNA sequence data. By incorporating the patient disease-specific variant, such as mutations or alternative splicing, it enables the detection of peptides that conventional approach cannot identify. Through this capability, we can discover novel biomarkers, guide patient-tailored therapeutic strategies, and even identify potential neoantigen candidate for immunotherapy. In essence, the proteomics shift from reference-based search approach to disease-driven approach, providing more accurate representation of patient-specific biology and deeper insight into disease mechanisms. So next topic is evaluation of biomarker using clinically available samples. Extracellular vehicles, or EVs, carry rich molecular cargo from tissue into blood and other body fluid, providing a lot of information from tissue that would not normally require direct tissue sampling. So EV profiling allows us to study drug mechanism or disease state without a biopsy. In our workflow, EV are enriched from blood or conditioned media and analyzed using our multi-omics capabilities. So for using the immunoaffinity purification, we can also capture the isolate tissue-specific EVs, such as brain-derived EVs, enabling the identification of translational biomarker for clinical studies. So our next example is a target engagement directly in humans. We utilize activity-based protein profiling, or ABPP, on blood cells like PBMCs as a target tissue. ABPP uses chemical probes that covalently bind to the active site of enzyme, allowing us to enrich and profile specific enzyme families. When combined with drug treatment, this approach enables us to assess both on-target engagement and the potential off-target risk at the same time. applied to PBMC from clinical study participant. ABPP provide data to support exposure response monitoring, dose justification, or proof of mechanism in individual patient. So next is the final slide. The key message of our presentation is multi-omics makes drug discovery more human relevant and more predictable. By focusing on mechanism first, we can reduce risk, accelerate decision making, and bring stronger biomarker into the clinic. Our platform integrates multi-omics acquisition, analytics, and interpretation into single workflow. In addition, our discovery-oriented proprietary platform helps to translate unknowns into the actionable narratives. And also applied layers such as EVs and ABPP-based patient target engagement allow us to connect the bench result directly to clinical readout. Okay, that's all. Thank you for your time and attention.
Thank you, Yoshinori. We have 4 questions here all ready and set to go for you. Our first question tonight, what is your roadmap to develop extracellular vesicle biomarkers?
Okay, thank you. So our roadmap depends on the intended application. So one approach is disease monitoring. In this case, extracellular vehicles can be used to measure tissue function. For example, liver-derived lipid detected in ABs can serve an indication of liver function. In that case, AV derived from the liver in diseased patient will be applied to proteomics and biomarker analysis to discover appropriate biomarkers, then applied to clinical studies. Another application is confirming of drug mechanism of action. Since drug response biomarker originating in the target tissue can be monitored through the extracellular vehicles. In this case, candidate biomarker are first identified in preclinical studies, and then we can get the molecular signature of the drug action. In both cases, we perform analytical validation of candidate biomarkers using the blood samples to prepare for a clinical application.
And thank you. Next question. When a novel disease-related molecule is identified, how do you translate it into drug discovery?
Okay, thank you. So in that case, we begin with a hypothesis generation from the findings defined in the molecule biosynthetic pathway, and also predicting the cell types and disease-related conditions. We then evaluate biomarker validity, both analytical and biological, as well as the therapeutic potential, considering factors such as durability, causability, and safety as well. And finally, we integrate the finding into the practical proposal and outlining the risk and supporting data and recommend experiment study so that the discovery can confidently progress to drug development.
Okay, very good. Third question for right now. How do you ultimately judge off-target risk for nucleic acid medicine?
Okay, thank you for the question. In that case, we assess off-target risk using 2 complementary approaches. First, we evaluate differential expressed genes considering both the number and the magnitude of changes, and also apply it to the mechanism of action analysis, such as principal component analysis. This approach can be used to confirm the quality and gene expression data quality of gene expression data. And we can highlight the potential differences in mechanism of action among the candidate. And then second, we conduct the function-based evaluation by integrating genome-based prediction with transcriptome data to identify candidate off-target genes. Their function and pathway are analyzed, and we cross-reference them with the risk gene list provided by Japanese regulatory authorities. Finally, our in-house safety expert reviewed the evidence to assess the clinical relevance and the security.
All right, very good. And our last question for this segment, Yoshinori, what do you do when different omics layers show conflicting results? For example, mRNA changes, but the protein does not?
Okay, so that sometimes happens in multiomics analysis. And in that case, we interpret the discrepancies by considering the biological hierarchy, such as genes and proteins and metabolite, and the time lag inherent to each layer. So messenger RNA often respond earlier than protein level. In that case, protein and the metabolite changes following depending on the translation and degradation and the pathway and the kinetics. So to clarify inconsistencies, we perform targeted validation, such as protein quantification by mass spectrometry or ELISA, and also gene expression confirmed by qPCR. By this confirming process, we can assess what is the hierarchy of each omics layers.
Thank you, Yoshinori. That was an excellent segment. Let's now bring in our third speaker. Joining us now is Jason Hamlin, PhD. He's a consultant for Vitrovo Incorporated. Welcome, Jason. Let's begin.
Thank you very much for having me. I'm glad to be here today. So we've learned that Axcelead drug discovery partners are an integrated drug discovery provider, and they have a global reach. Plus, Axcelead has embraced the new approach methodologies. For Vitrovo, they are highly specialized at in vitro neurological assays, especially microelectrode arrays, which are helping to build industry standards on how MEA data can be used for drug discovery and toxicology. They've built a team of more than 10 scientists and can deliver reports extremely fast in as little as 2 months. So by working together, Axcelead and Vitrovo connect broad drug discovery capabilities from Axcelead with specialized neuroscience expertise from Vitrovo. So it's great to see these companies collaborating constructively to move NAMs forward. Just a quick outline of my presentation. First, I'll talk about the new approach methodologies and how MEA data supports them, including expertise in data analysis and AI-based compound identification and mechanism of action prediction. So regulators are increasingly supporting new approach methodologies, or NAMs. NAMs are modern testing strategies that move beyond traditional animal studies. They include in vitro human-based systems, computational models, and other innovative platforms like microphysiological systems, or MPS, designed to evaluate safety and efficacy more efficiently. So on the right, you can see a screenshot of the FDA's roadmap for reducing animal testing in preclinical safety studies. This was released in April of 2025, and I highly recommend a review. It's a concise 10-page document that researchers can learn more about NAMs and how 18 other US agencies are aligning to accelerate adoption. It contains handy information on how the NIH is aligning and how that could influence things like grant funding for projects. Another key body just below is the OECD, which plays a central role in harmonizing testing methods internationally. So one example is their guidance on in vitro assays for developmental neurotoxicity, an area where MEA technology is directly applicable. So I'll talk about developmental neurotox in a couple of slides. But first, just so we were on the same page, what exactly is MEA? Microelectrode arrays measure extracellular voltage changes from groups of excitable cells such as neurons and cardiomyocytes. Now, these readouts can reveal risk for seizure liability, for example, identifying network hyperexcitability. And we'll look at some examples of that. Just for context, EEG actually has been around for nearly a century. So Hans Berger first published on it in 1925, so we're at the 100-year anniversary. But that captures organ-level activity, the whole brain, and lacks sensitivity. Patch clamp is exquisite at its ability to measure detail, but it doesn't really measure network activity. So MEA on the right there sits right in between, and it appears perfect for recording networks of cells with speed and reproducibility, and you can use things like iPSCs and organoids with it. So it works for NAMs. And in fact, MEA provides a lot of data, but how is this data really mapped onto what we're talking about with those regulatory assays and parameters? On the right, you can see the OECD guidance document for the developmental neurotoxicity in vitro battery, or the DNT-IVB. The DNT-IVB includes assays such as cell death, migration, neurite outgrowth, and critically, it has network level effects. And that's where MEA really supports the OECD NAM model. So functionally, compounds with excitatory or proconvulsive effects drive more frequent, taller, and tighter bursts. Depressants flatten the network activity, increase irregularity. So you can see there's a lot of parameters you can measure. This is done longitudinally without destroying the cells. So you can get, in fact, a lot of data using the MEA for this purpose. And in fact, Vitrovo offers unique expert data analysis. So on the left side of the screen, you can see how compounds can be classified into low, medium, or high seizure risk, depending upon their distance from the baseline. On the right side, you can see a correlation to in vivo dose. Now, this is important as the FDA roadmap is asking researchers to provide a correlation between in vitro NAM assays and animal models. So in that assay on the right, basically, you're looking at the concentration of compound in the MEA dish plotted against the concentration of the compound in the CSF of the animal at tumor onset. And the data shows that the MEA network activity increases as the animal appears to tremor. So there's a strong correlation between the MEA data and the CSF data. And Vitrovo is actually applying these methods right now in a multi-site analysis led by the HESI consortium. And they also hold patents related to this methodology. So Vitrovo really is making the most out of this extensive data provided by MEA. And in fact, all of this data can also be used for AI-powered mechanism of action and compound identification. So in this example, raster plot images from MEA-treated cultures are used to train the AI. This is done in time segments and fed into the input. And it can be used to predict compound identity with high accuracy, greater than 99.9% on trained compounds. Spike trains are visualized as raster plots, segmented into the time windows, and then passed into a convolutional neural network. The network extracts features such as spiking patterns, and then outputs predictive labels such as mechanism of action or compound identity. So not only by leveraging these large data sets, we can actually combine that with the power of AI to achieve accurate compound prediction and mechanism of action prediction. So that wraps up the section on new approach methodologies, but let's talk about ultra-high density MEA. What exactly is ultra-high density MEA? Well, the state of the art now shows that ultra-high density MEA is a large single well with 236,880 electrodes. That's at least 50 times greater than the next nearest technology, which is called high density MEA. The sensing area, which is 5.5 by 5.9 millimeters, is larger than most other wells, large enough to fit an entire brain slice, as you can see on the right. Cortex and hippocampus, where you can watch propagation occurring right through the tissue. So currently, this UHD MEA is available exclusively to Vitrovo and selected partners, and you can contact them for more information. And so what this resolution offers is the ability to see connectivity in a way that's just not possible with standard MEA systems. So I'll show you how this applies to organoids. Now, if you look at the left here, this is a human neural organoid that's plated on an Ultra HD MEA dish. And you can see connection analysis at basically the single cell level. And what's remarkable about this is that all of the electrodes are recording simultaneously. so that spikes that occur within a 100 millisecond window are considered connected. And that allows us to build a detailed connection map. So on the right side, you can actually see how this map looks. The top panel shows the baseline where connection scores form a tighter, more synchronous peak. In the bottom panel, after addition of picrotoxin is a GABA inhibitor that disinhibits the network, you can see activity increases, but it's spread out with more connections forming among neurons and less of a tight synchrony seen in the baseline. So this Ultra HD MEA makes it possible to run extensive connection analysis and mechanism studies that go far beyond what's possible with existing MEA systems. In fact, we can use Ultra HD MEA to measure axonal propagation velocity. So on the left, you can see a neuron firing and the signal propagating along its axon. And this is, I think, over 4 milliseconds. We can trace the same axon before and after treatment to discover the true velocity. Now, this is a direct recording of a single cell firing along its length. On the right, when the axon or the cells are treated with vincristine, a microtubule targeting anti-cancer drug that is known to cause peripheral neuropathy, we can actually see neuronal conduction increase transiently after 2 hours. And the exact mechanism isn't fully clear, but the effect is strong. Interestingly, by 24 hours of washout, the velocity recovers to baseline. So this type of assay provides a direct functional readout of axonal health, allows us to measure compounds that impact neuronal communication. It adds to weight of evidence for compound safety and profiling. And honestly, it's pretty remarkable just to watch axon propagation in real time. And so we can actually use Ultra HD MEA for the creation of pain, sensation, microphysiological models, or MPS. These organ-on-a-chip systems are increasingly being adopted into the NAM framework. And there's a huge unmet need here, of course, safer pain medications. So on the left, you can see a simplified schematic of the pain pathway. Sensory neurons project to the dorsal root ganglion and then into the spinal cord. From there, signals are relayed upward to the brain. Understanding how the relay works, and it allows us to be able to interrupt the relay. And the goal is to block transmission, of course, before it reaches the cortex. On the right, you can see components of the microphysiological system. So we can combine microfluidic chambers with iPSC-derived sensory neurons and iPSC-derived spinal cord cells, and we can reconstruct pieces of this pathway. And so we'll be putting it on the Ultra HD MEA. That's the device pictured on the right, and assay this in real time. So how does this look when we actually put this on the system? So in the top left, you can see the setup. We have sensory neurons plated in the left chamber, their projections extending through the middle channels, and iPSC-derived spinal cords in the right chamber. So in the left movie, you can see sampled images every 50 milliseconds, and this is slowed down slightly just so that you can see some activity, some baseline activity from the sensory neurons, a little bit of activity projecting over to the spinal cord, and the occasional spike appearing in the spinal cord. What happens, though, when we add an agonist, or basically AITC? This is a compound which gives mustard and wasabi that delicious spicy flavor. And of course, our sensory neurons react to it. When we play the movie, we can see an intense reaction from the sensory neurons. But if we watch carefully, we can see the propagation through the channel, through the MPS chambers, over to the spinal cord cells. And what we see is even after the sensory neurons have stopped firing, we still see this residual activity in the spinal cord. So in fact, this type of network propagation, being able to interrogate the network propagation over time, really allows us to test novel and new drugs to interrupt this process and hopefully find new non-CNS effective pain medications. So in conclusion, here's what Vitravo offers. They really provide cell-based assays to customers worldwide. They have culture know-how, the ability to set up MEA and in vitro models, and have access to the Ultra HD MEA system. They can run a variety of assays. Today, we focused on MEA, but they cover a full range of cell-based in vitro neuroscience assays to support assays like the DNT IVB that I mentioned. And they combine this with that expert data analysis for compound risk assessment, onset dose prediction, and AI modeling for mechanism of action identification. They've already worked with 8 out of 10 Japanese pharma and are expanding globally. They run over 30 projects a year and can deliver results in as little as 2 to 3 months. Their approach is agnostic, so whether you're working with peptides or antisense oligos or RNA, they can support your research. So with that, that concludes my section of the presentation. Thank you very much, and I'm happy to take questions.
Thank you, Jason. We have a number of questions here. Why is MEA data especially relevant now in the context of regulatory guidance, such as the OECD's developmental neurotoxicity framework?
Right. So really, regulators are looking for more robust, I think, in vitro systems that can supplement and eventually replace animal testing. The guidance has a lot of different assays. So there's like, neural migration, division, cell death. These are more sort of classical assays, but they also ask for network activity, things like spikes, bursts. Obviously, this is because of the risk of seizure or drowsiness or coma risk in neural indications. So by aligning the MEA with the OECD framework, we're able to provide that data in a more standardized way, reproducible across labs, and relevant for the framework.
All right, thank you, Jason. Another one here. Ultra-high density MEA technology sounds impressive with over 236,000 electrodes. Can you please explain what this scale enables that traditional MEA platforms simply could not achieve?
Right, so so how many, do the electrodes matter? In fact, we appear to have crossed a threshold where we're not just doing higher resolution, we're doing assays that just were not possible before. So you saw the propagation or the single cell axonal speed propagation, where we saw the multi chambered sensory to spinal propagation. That's like 50 to 200 times the scale of other MEA platforms. So that allows us to map the full network dynamics at once, map the synaptic connections, and generate hundreds of parameters per neuron. So this is basically not just does what is existing better, but opens up a lot of new assays.
Very good. Let's see here. You've also shown examples of AI analysis and even microphysiological models for pain and sensation. How close are we to bridging these advanced in vitro systems to actual in vivo outcomes in drug development?
So this is a great question. This is where Vitrovo is pushing the field. For example, by training the AI models on the raster plot images, they've been able to predict mechanisms of action and identify drugs with 99.9% accuracy. So I guess the idea is we're not claiming to have replaced animal studies yet, but we're on course. The combination of the Ultra HD drives the advanced analytics, and that drives the AI analysis, giving us sort of a path forward. And that's sort of the essence of in vitro to in vivo extrapolation. So I guess my answer is I think we're pretty close and on track.
Great. Thank you, Jason. Let's bring Masayuki back in to wrap up today's session. Masayuki, take it over.
Okay, thank you all for joining today's webinar. As we've seen, NAMs are transforming translational research by enabling the human relevant predictive models. We hope these insights help accelerate your own innovation. Please feel free to reach out if you'd like to explore collaboration. Have a great day. Thank you very much.
Masayuki, before you log off, we have one final question.
Yeah.
Beyond your own platforms, what other promising NAMs approaches are being explored in the field?
Okay, thank you very much. Great question. In addition to our platform, there are several existing NAMs initiatives globally. For example, many groups are advancing MPS systems, MPS using IPS such lung tissues. These approaches complement our work and reflect a broader movement towards human relevant integrated in vitro models that can reduce reliance on animal testing while improving translational predictability. Thank you.
Thank you, Masayuki. And that's all the time we have today for questions. If anyone submitted a question that wasn't addressed, keep in mind that the speakers will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And before you log off, please take a moment to complete the feedback form so we can continue to improve your webcast experience. On behalf of EBD Group Bioeconomy, part of Informa Connect Life Sciences, thank you and have a great day.